Hello everybody, my name is Roman Bauer and today I'm going to talk about different machine learning based methods for understanding and predicting brain health. First of all, I would like to thank the organizers for having me. So in this research, we are interested in the brain, and the brain is, as you know, a very complex organ. The human brain has approximately 100 billion neurons. Here we see a visualization of brain fiber tracts as achieved with diffusion tensor imaging, which measures the diffusion of water molecules in the brain. And here you can see with different colors indicating different orientations, those fiber tracts. And what is immediately clear is that this is a very complex organ. And so we need computational methods and we need machine learning to gain insight into for example, if some of those fiber tracts are different from the healthy state, or we can also employ machine learning methods on other aspects of the brain, including activity. And what I would like to highlight here initially is that there are different approaches, computational approaches to achieve this. Some of the approaches are more statistical. So here on the left-hand side, we have approaches like, for example, deep learning based approaches and modern AI based approaches, which are usually more data driven. And then we can also look at this kind of work from a more model-based perspective, where we have really models that go uh, directly towards modeling those individual processes themselves and relying less on creating the modeling from the data initially. And so there's a lot of background knowledge that goes into this kind of model-based approaches. So I would like to a little bit delve into this, and as a first approach or a line of research where we have used machine learning for predicting or diagnosing brain health, we have here used EEG in this paper here that we published a few years ago. We have used machine learning to assist in dementia diagnosis using EEG data. And one important result that we discovered here is that there is a big difference in healthy subjects. So HC stands for healthy control subjects. When we look at the dominant frequency variance of eyes closed versus eyes open EEG signals. So on the y-axis here, on the left-hand side of this plot, we have this ratio of this DFV feature, and there this ratio of eyes closed versus eyes open is usually quite high. So you can see here around up to like 8 almost in healthy controls, but in Alzheimer's disease subjects, this ratio is much lower and significantly lower, statistically validated, as you can see here. And so here on the x-axis, these are different brain regions, and the G stands for global, F for frontal, and so on. And we have in different parts of the brain, we can see or we can observe this difference with regards to the ratio of eyes closed versus eyes open for the dominant frequency variation, which is significant, as I said, between healthy controls and Alzheimer's disease subjects. And so this shows that actually looking at EEG signals is not only useful, but highly informative also for diagnosis of dementia. We have also looked into other dementia types. And what is also very important is that we should also consider eyes closed and eyes open, because often research studies focus on one type, either eyes closed or eyes open. But here we show that the eyes closed versus eyes open ratio of this particular feature is highly informative. And so that is one of the results of this paper. And then by using advanced machine learning methods, we could actually also classify other dementia types. So AD stands for Alzheimer's disease, DLB is for dementia with Lewy bodies, and Parkinson's disease dementia is PDD. And you can see here that our classifiers performance in differentiating these dementia types is significantly high with very good sensitivities, specificities also. And so this is a proof that EEG signals can be highly useful for dementia diagnosis. And what is also important is, as I said, is we should ideally also include eyes open as well as eyes closed in order to really get more information out of those type of recordings. And what we have not done there, but which is something I would be very interested to do in the future, would be to also analyze how those signals vary at different locations, spatial locations, and then try to better understand also the temporal changes during the evolution of those brain disorders or dementia types. And so in a paper that was published several years ago, people have looked into EEG based analysis with aging, so the impact of aging on this, and they have also investigated how different features depend on spatial locations on the head. And so this is something we could also potentially in the future look into more to really understand then also spatially what is happening with the pathology, how is it evolving over time. And so this is a very exciting research direction to go into in the future. And this is actually quite relevant to another study that I want to a little bit discuss here with you, which uses diffusion tensor imaging. So previous papers have shown that one can use data obtained from diffusion tensor imaging to create, for example, connectivity matrices. So we can first parcellate the brain into Brodmann areas, and then we can employ methods called tractography methods in order then to sample and create virtual models of those fiber tracts that I showed you at the beginning of this talk. And then we can mathematically infer connectivity matrices and ultimately brain graphs from the DTA data. And this allows us to create an abstract representation of physical connections. So what I showed you before with the EEG were activity patterns, but here we can actually also look into estimates of connectivity between brain regions. And in this paper here that we published a few years ago, we show that such brain graphs can also be very powerful in order to detect Alzheimer's disease, and potentially very early on, even earlier than what is currently possible in a clinical setting. So what did we do in this paper? So we had two different types of data. We had control data and Alzheimer's disease data from DTI, and then we used machine learning to create a predictive model. And we essentially leveraged a model that had stages of disease progression. So we modeled effectively how the normal aging process changes the brain connectivity. So this would be this side here, this arrow here. And then we also modeled how Alzheimer's disease changes the brain connectivity because these are different types of changes. Obviously, there is a lot of changes happening during brain healthy or normal brain aging, and there is also a lot of changes happening during Alzheimer's disease aging. And so, what we wanted to do is we wanted to create a model for each of those in order then to early on identify where they significantly deviate from one another in order to potentially create an early diagnostic method. So, as I said, we looked into brain graphs, and one very powerful methodology to make sense and quantify brain graphs is um, based on complex network properties. So, we essentially have a graph, and then we can associate, for example, strengths with individual connections. This is also something we can infer from diffusion tensor imaging. And some of those nodes have more connections and stronger connections than others. And we can use this type of information to quantify various aspects of brain graphs. And for example, we can look into features like betweenness, centrality, or closeness, or page rank. So these are all measures that reflect the centrality of certain um, graph properties. And what we identified is that those centrality related measures were very important to differentiate healthy from Alzheimer's disease brain networks. So you can see here that those percentages are quite high for all of those complex network properties. These are all centrality related complex network properties. And we then use those properties to train machine learning methods to classify Alzheimer's disease versus healthy at different stages. So remember, we simulated the progression of those diseases and the normal aging changes. And as you can see here, the classification performance increases with stage, which is not surprising because the more evolved the disease has, the more striking the differences are. And so that's why you see here this increase in those performance measures with stage. Notably, a stage here is unitless, but it approximately corresponds to 1 year. So we modeled essentially how the brain connectivity changes across 15 years of aging. We did this because we know that Alzheimer's disease is usually a very slowly progressing disease. Post-mortem studies have actually identified Alzheimer's disease pathologies even in subjects in their 30s. So it is very likely that this is something that starts early on in life, but we don't notice it until very much later. And so what we show here in this model is that actually with this stage-based model, we have evidence that our DTI-based diagnostic performance is increasing and then gets very significant. So here you see a p-value of 5%. So you see here that the -4, it is very low. The p-value is very low. And so we have high statistical significance at -4 stages, which is approximately -4 years from current clinical diagnostic performance. And so this is evidence that using DTI could potentially help early diagnosis of Alzheimer's disease. And as you may know, some of there are already some medications that seem to slow down Alzheimer's disease progression. There's not yet a cure, but at least there are some drugs that seem to slow down the overall progression. Now, another modality that we looked into is retinal fundus images. So we have looked into uh, analyzing with, again, in an automated manner with machine learning and computer vision methods into analyzing retinal fundus images. So the retina is a neural tissue at the rear of the eye. And this is a very practical type of modality for diagnosis because of several reasons. So one reason is that it's a very simple technique to master. So taking those photographs is actually quite simple. It's much simpler than having requiring a diffusion tensor imaging based measurements or MRI measurements. So you need much less training. It's very cost efficient. It's very cheap. You can actually buy cameras for quite low prices, and then so just taking a photo is not expensive at all. It's very quick. Like an MRI scan can take an hour or so, and here this really takes seconds. It's non-invasive to take those fundus images, and that's also why it usually has very high patient compliance. So usually, also for elderly, it's usually quite straightforward to obtain those images. And because of those practical benefits, it's also possible to monitor over time. So we can take those images, for instance, every month or so, in order to see how things evolve over time. And previously, there are several studies that show that one can use those retinal images for detecting many diseases, not only diseases of the eye, but also systemic diseases, including, for example, heart disease related risks. And so what we did is we actually created a more explainable methodology to detect neurological conditions. So there's rather limited insights on the use of retinal images for neurological diagnosis, but we showed actually that we can indeed analyze retinal fundus images with automated methods, and we do this by segmenting the vessels. So in retinal fundus images, one can visualize the vessels, the veins, and the arteries. And we can segment this with computer vision methods, and then we can classify into arteries and veins. And we have published previously papers where we show as a proof of principle that indeed we can then extract features from those arteries and veins and then train machine learning methods to classify whether an image is likely from a person who has a neurological condition like Alzheimer's disease or Parkinson's disease or neurovascular dementia, or on the other hand, does not have any of those. And we did this, as I said, by classifying, at first segmenting and then classifying the vessels. And then we had also used methods where we have concentric circles and then we extract the features for those different circles. So here you see the different features that we employed. In this case, we had nine features, and then we associate those with specific zones, so which are those specific circles, and we then extracted for those zones those particular features, and they were informative for neurological diagnosis. Here you see a more recent paper that we published just last year, and this was published at IEEE conference on AI, where we then even show that employing certain machine learning methods and AI methods that are very supportive of explainable hypothesis generation. So they allow to generate also hypotheses that can be understood by humans, non-experts, and they don't need to be computer scientists. And our method PyGoal that our student Daniel Cyrus used here performs very well, even with quite a limited number of examples. So we get really very close to I think 95% or so at around 90 examples only. This is an important criterion because usually there are not many data from Alzheimer's disease and Parkinson's disease that we can use. So it's important also that we are efficient in terms of samples, and most deep learning based methods are not very efficient in terms of samples needed to train those models. That's why large language models need insane amounts of training data. They need very large training volumes. And so here we show that in Alzheimer's disease and Parkinson's disease and also neurovascular dementia, we can achieve very high performance by employing this inductive logic programming type of AI called PyGo. And you can read more here. As I said, PyGo also allows to infer hypotheses. So it allows to create hypotheses that then could be tested potentially experimentally by clinicians. For example, here this is just to give an example of how such a hypothesis looks. So it could, for example, infer that if the tortuosity of the artery is higher than a certain value and the tortuosity of the vein is lower than another value and so on, that then this person has Alzheimer's. And so this really gives us some hypothesis that would be very difficult to create just without any AI, right? So it's really very helpful, I think, for accelerating the process of generating hypotheses. So overall, I've shown you today that we can use computation models from different kind of types. So some are more data-driven, some are more mechanistic. Um, for example, the DTI-based method is more mechanistic because we there really modeled how the pathology spreads, right? And then there are other models which are more data-driven. And then we have also explainability as an important criterion for this kind of diagnostic tools, because it's very important that we understand why a tool gives a certain type of diagnosis. Because often then, if we prescribe a drug, um, and we need to understand why this drug is recommended. Obviously, there are often many side effects, right? So we need to really understand diagnosis. And and I've shown you examples where we use MRI and DTI, EEG, and retinal imaging data to diagnose neurological conditions. And I'm very interested in continuing in this line of research. So if you are interested as well and have maybe some relevant expertise and data or other maybe suggested approaches, then please don't hesitate to get in touch. So thanks very much for your attention. And if you should have any questions, please don't hesitate to let me know. And I wish you a nice rest of the conference.